life cycle and molecular mechanism of quorum sensing in Myxobacteria. So let us start with this particular topic. Now, this is basically about how the bacteria communicate with each other. Now, you know that higher animals can communicate with each other, but even the lower animals can communicate with each other, and they show what is known as behavior. Like for example, honeybees also communicate with each other. You must have learned in zoology about how bees communicate with each other. So this is the basic communication which takes place in prokaryotes, and it is known as quorum sensing. So we will be discussing about this, and studies with these organisms which can be used as model help us to understand how it is possible for higher eukaryotes. So eukaryotic cells also communicate with each other, or they also show social behavior. So eukaryotes, higher eukaryotes are known to show such social behavior, but this social behavior is also seen in prokaryotes. So we are going to see some examples here wherein various kinds of bacteria show social behavior. Now, what I mean by social behavior. So, let us first see a little bit about Myxobacteria, and then we are going to go into details of how they show social behavior. So, Myxobacteria are basically gram-negative bacteria. Most of them they grow as terrestrial bacteria. Some are formed as marine forms, and these bacteria, they are basically saprophytic, or sometimes they are hyperparasitic to other bacteria, wherein they produce hydrolytic enzymes, and they can feed on other bacteria. So they can lyse these bacteria and feed on them. These bacteria usually show slight variation in shapes. Most of them are rod shaped, so gram negative rods, but they can produce resting structures which are known as myxospores, which are spherical. They show social behavior, and the cells communicate with each other on in a cell density dependent manner. So the communication depends upon the cell number, and this communication is through small signaling molecules, and these signaling molecules are secreted out by the bacteria, and which are received by the other nearby bacteria of the same genus and species, and also by the same cell. As I mentioned just now, these bacteria, they are predatory in nature, so they can produce hydrolytic enzymes, which include cellulases, chitinases, proteases, and they also produce broad spectrum antibiotics. So this is a very important phenomena or important property of Myxobacteria, because of which they are largely studied. Because you can study Myxobacteria because they produce novel antibiotics. Antibiotics which are not studied till now can be discovered by studying Myxobacteria. Now, about the morphology of various types of Myxobacteria. So in Myxobacteriaceae, genus Myxococcus is the most well studied genus, and we are going to see the life cycle of Myxococcus xanthus, which is the most well studied species with respect to the life cycle and other phenomena related to morphogenesis in this particular organism. So they are these Myxococci, they are rod shaped cells, but they produce spores which are spherical, and hence the name Myxococcus. They produce most of the Myxobacteria, they produce slime, and which is also known as EPS or exopolysaccharide. And this slime is very very essential for the motility of Myxobacteria. uh, the spores are produced in sporangia, and which may or may not contain stalks. Arcangium is another genus which has rod-shaped myxospores. Then there is a genus which is known as Cystobacter. I'm going to show you pictures later on of all these representative genera which I'm discussing just now. So it produces fruiting bodies without stalks. Stalk fruiting bodies with single sporangia are produced by Militangium. Then stigmatella is also one interesting bacterium. So it produces stalk fruiting bodies with multiple sporangia. So branching is seen in case of stigmatella. Then, in case of these polyangium, sorangium, etc., the vegetative cells are not tapered but with blunt and rounded ends, and the myxospores resemble vegetative cells. So, there is not much difference in the morphology of vegetative cells and the myxospores. Sporangia are produced, so fruiting bodies are produced without stalks. Myxospores are rod shaped in polyangium. Similarly, in sorangium also, fruiting bodies are without stalks and myxospores are oval in this case. They are highly cellulolytic bacteria. So I will not read all the information for all of them. Now, this is how they appear. So, because they produce spore bearing structures or fruiting bodies, which are which are lifted above the medium. So, if you take a photograph or from the side, you can see the fruiting bodies clearly. So, this is a fruiting body of Myxococcus fulvus. Then, the second picture, so the second picture is for the fruiting body of Myxococcus stipillatus. Then chondromyces. You can see that chondromyces shows branch fruiting bodies with so on every branch again spores are present in this sporangium like structure. Then this diagram here is for stigmatella. I told you that stigmatella also produces multiple spores. This is a fruiting body of stigmatella aurantiaca. Now you can see here the colors are real. So they are known to produce carotenoid pigments, and their colonies appear yellow or orange because of production of these carotenoid pigments. Uh, Visibly also you can see these colonies. Now these myxobacteria, they produce very flat colonies. They are not having elevation as such. But when they produce sporangia, then these sporangia are above the surface of agar. So you can see that they are lifted above the surface of agar. Now this here is a diagram electron micrograph of Myxococcus, the rod-shaped vegetative cell. You can see it is about to divide because there are two DNAs already synthesized here in this particular cell. It divides normally like bacteria by binary fission, and when it is growing vegetatively. Under the conditions of starvation, the differentiation will take place and the morphogenetic cycle is induced, which results into conversion of the vegetative cell into spore. Of course, I told you that it shows social behavior. So, if a group of cells come together, only few cells will get converted to spores and few cells will get converted to stalks. They will die, they will produce a lot of mucilage, and they will get converted to stalk. So the spores which the vegetative cells which get converted to spores, they get they become spherical in shape, and they will develop a thick outer covering. You can see here, as compared to the covering here in the vegetative cells, this is a very thick covering which is protective because these are resting structures which can germinate when nutrients are available. Now, I have taken these diagrams from Brock's Biology of Microorganisms.